Hi everyone. Hi everyone who is watching online and who will watch this recording. First off, we would like to begin today by acknowledging the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respects to their elders, past and present, and extend that respect to all Aboriginal and Torres Strait Islander peoples joining us today. I'm myself coming to you from the land of Jayabal and Jarawar people. And we actually invite you to share in Q&A box the lands that everyone who is participating today is working and living on, if you would like to do that. So today we're really lucky to be joined by, four really diverse panelists with diverse backgrounds. So first off, we've got here Professor Jeff Powell, if you can just maybe wave, who is the theme leader for soil biology and genetics with the Hawkesbury Institute for the Environment and at Western Sydney University. Then we've got Dr. Anna Hopkins, who is the senior lecturer in conservation biology and environmental science at Edith Cowan University. And Anna also sits on the ESA Plant Soil Ecology Research Chapter Committee. Then we've got Giancarlo Chiarenza, who is a PhD candidate at UNSW at the Big Ecology Lab. And Gian is also at the sits on the ESA Plant Soil Ecology Research Chapter Next Generation Committee. And finally, myself, I'm Dr. Christina Birnbaum. I'm a lecturer in terrestrial ecology at the University of Southern Queensland, and I'm the convener of the ESA Plant Soil Ecology Research Chapter. So just quickly before we begin, we wanted to note that this webinar is actually part of the ESA Plant Soil Ecology Research Chapter activities, and more specifically, it's aligned with the symposium, ESA symposium that's held between 3rd and the 7th of July in Darwin. And the symposium is called Unravelling the Underground, New Insights into Plant Soil Ecology from Diverse Fields. And so you'll see that there's a bit of alignment in the title of the symposium and this webinar. And I will just also share for people who might be interested in the symposium and the research chapter. So we've just put a slide up here to show you the details of the symposium and the conference that you can still register if you're interested, and also the contact details of the research chapter as well. We're also on socials if that's your thing, so give us a follow there and get in touch if you're interested. Okay, so thanks for that. So that was the intro, and now we'll begin. So first of all, thanks to everyone who have submitted their questions for this webinar. We've got quite a few to get through, so that's great. But if you also have other questions to the panelists, please feel free to pop them in Q&A, and we will try and answer them as we get through the seminar. Excellent. So we'll start off with a first question. So I'll just read the question out, and then I'll invite the panel to comment and answer. So first questions, are there good examples of how interdisciplinarity across plant soil ecology disciplines has led to advances for fundamental ecology and applied outcomes? So I'll invite maybe Jeff to comment.
Sure. Yep. Thanks, Christina. When I think about that question, I think back to some of the foundational work in soil ecology that took place at back in the 80s. There were papers published back then. People who were trying to get a good understanding about what happens to carbon and what happens to nutrients once it ends up in soil, and starts to cycle around down there. What are the organisms that they end up in? Like people knew about earthworms, people knew about microarthropods, people knew about fungi and bacteria, but trying to get an understanding about how those food webs are linked. And so they would do studies where they would add antibiotics of certain kinds to soil, and study changes in population dynamics of these different groups of organisms, and look at changes in available nutrients and so forth. And so there was a lot of biogeochemistry involved in that. There was a lot of taxonomy involved in that. But there was also a lot of math involved in that. It was really foundational from the perspective of getting a strong understanding of how ecological communities and food webs are structured, and what makes them stable, and under what conditions can they start to collapse. And yeah, so that's and that's maybe something that we don't do enough of currently, is thinking about theory, and can we use math to try to get a better understanding of, in general, about how our soil systems work.
Really good point. So, Jeff, are you suggesting that sort of in the 80s and back then, the soil or plant soil ecology was actually quite interdisciplinary?
Oh, it absolutely was.
Or yeah, and it's still maybe is to some extent, but yes.
Oh, it certainly is. But we just tend to focus on interdisciplinarity in different ways. So we've got molecular biology is a huge part of what we do now. So we haven't lost that. I'm not saying that we've lost that. We say we can continue to gain from some of those disciplines that we did work with a lot.
Mm-hmm. Yes, definitely since the sort of 2000s, the onset of the high throughput sequencing has definitely opened what we used to call, the black box below ground. Definitely has helped to shed light below ground, and then yes, link that with the above ground ecology that we know. Actually, when you were talking, this reminded me when you mentioned theory, Jeff, that at the symposium that's going to be held next week as part of ESA, there will be a lot of talks that are actually tapping back to the theory. So taking the trait ecology that, is very well used and applied in a sort of plant ecology, focusing on the above ground, so we know the leaf economic spectrum and other things like this. So there'll be a lot of interesting talks actually that then try to apply a trait ecology and framework to say, size of fungal spores or fungal traits or microbial traits. So that's really exciting. I think sort of interdisciplinarity that we maybe haven't had that much, at least definitely at the symposia. So that's really exciting. Excellent. Anyone else has any comments on that?
Kristen, I'm happy to just jump in for a sec. One of the things that I think is really great about soil ecology is that it really does lend itself to that interdisciplinary focus, because, I'm a little biased, of course, because I'm a fungal ecologist, but I always think that soil is kind of the key link between a lot of those different components of the ecosystem. So you can see great examples in the literature of obviously people who are looking at that link between, say, what's in the soil and what's happening with the vegetation and stuff, but then you can also link it, say, to animals that are macrophagous, or there's a whole lot of nice links that can go on in there in lots of different ways. So I think that as a soil ecologist, you have to be really interdisciplinary. Partly originally, I think that was in a way to justify our existence, to say, yes, if you understand what's going on in the soil, that helps you to then understand, what's happening with nutrient cycling or what's happening with the vegetation or something like that. But I think it's becoming recognized more and more now that those activities in the soil are really fundamental to a lot of the other activities in the rest of the ecosystem. And so that does mean that we have to think about the relationships and the links between those other parts. So
definitely. Yes, I think recently there's been this push to include, soil biodiversity into this One Health framework with United Nations, where, we're just acknowledging the important role soils play, not just in, natural ecosystems, but also urban ecosystems, and how that all is linked with human well-being and human health as well. So promoting that more in interdisciplinarity, sort of the urban planning even, and human health, and not mentioning things like restoration ecology, where we're already doing a lot of sort of applying the sort of plant soil ecological frameworks and ideas to improve applied outcomes as well. So yes, definitely, I think as everyone has said, the soil, we're acknowledging it more and it's getting more traction across different fields. I feel the same. Excellent. Yeah.
Christina, just one other thing to add. There was a really interesting study that I've come across recently that some of my colleagues are doing in the research centre that I'm in, and that is linking education with soils and understanding. So they're actually using soils and children's exploration of soils as a way to engage them with their environment, and also to understand, you know, more about the soils themselves for those students. So that's another, like, it's a completely different interdisciplinary. So really actually interdisciplinary because it's education and, you know, sociology and things. And so that's a really fascinating extra example. I think that soils lend themselves to becoming accessible to those other disciplines as well, because, you know, everyone can. Hi, Indy, that's you I'm talking about. So, I think that's a really interesting example of that interdisciplinarity too.
Great example, and also tapping, you know, into the next generation and different, you know, demographic groups to explore soils and start to include that early on already in education, which is great. Excellent. Okay. We have another question that was asking, are there any disciplines that plant soil ecology researchers are not currently collaborating enough with, and if resolve would help advance these? So any particular fields? We already mentioned a little bit sort of urban planning and human health. And of course, there's agroecosystems and sort of understanding better our crop systems and what microbial communities, you know, they have either endophytes or fungi or bacteria, and how we can then use that to improve our crop production. But does the panel think there's any other fields that maybe we don't really think that much usually that could benefit from, you know, plant soil ecological sort of knowledge and expertise?
I'll just, so speaking a little bit from ignorance, and I could be wrong. I wonder, there's a lot of, I wonder about the field of economics. So there's certainly companies that are interested in using organisms that are isolated from soil as inoculants. And so there's an economic, there's certainly work that's gone into the economics of producing and distributing and using microbes in agriculture and restoration. But I wonder about more so from the perspective of natural capital. There are markets out there where people trade carbon credits. There's potentially markets that are going to be out there where people trade biodiversity credits. Or I guess those markets actually already exist when it comes to planning. How does soil ecology fit into that? How do plant interactions with soil organisms fit into that? Yeah, that's something that I don't know a ton about, but I think would be really interesting to find out more about it if it's out there, or if not, like I think that's probably something that we should be putting some focus on.
Definitely. I think. Oh, sorry, Anna, you go.
No, I was just saying it's a really interesting point, Jeff. I guess in the context of like carbon credits and carbon sequestration, there's already a bit of work in that area, particularly in the agricultural sector. But you're right, there's not so much in terms of biodiversity. You know, the focus really is solely on that carbon activity.
And I think we're just,
it's a real gap.
Sorry for speaking over top of you, Anna. I think we're still trying to figure out, like, how are soil organisms and their interactions with plants contributing to the building of soil carbon, and less so trying to, I guess we need to understand that before we can then focus on the you know, the financial aspect of that, or the way in which those organisms might fit into programs that that are trading those credits.
Yeah, I think these are great examples because I think, two things that sort of stand out to me is currently when we have any sort of credits, it's biodiversity credits, they're possibly mostly focused on the above ground, right? So plants, animals. But if we maybe applied some sort of index or biodiversity credit for below ground, you know, how diverse is your soil? Does it have all the important functional groups in the soil under that vegetation? That would be really neat, I think, and would promote possibly, you know, that sort of taking care of the soil. But when you were talking, Jeff, it reminded me when you mentioned economics that there's a lot of research that sort of went the other way around, applying economics to, say, bacterial dynamics and microbial dynamics generally, which is really cool. So there's definitely that interdisciplinarity, coming back to the previous discussion point, using, you know, other fields and applying to microbial ecology. And maybe that's another thing that we also could think in a reverse pattern almost, as opposed to where plant soil ecology could, you know, fit in with other disciplines, where we could maybe as plant soil ecologists draw on from to include and, you know, test some hypotheses as well. I'm
keen to get the next generation's input on this. Gian, what aren't we doing?
I think there is like slowly coming up is when we talk about ecosystem services, there is a lot on how we should quantify these services. A lot is on always plants, above ground biomass. The potential to sequester carbon, let's just give in some dollars, some value. Soil, almost never there. It's still able to quantify carbon. There is still biodiversity. However, it seems like this big black box where everything that is not on plants goes there. And I think like we should collaborate more, because I think we should be able to try to like disentangle all these aspects, because I think soil provides ecosystem services that are like almost as much as what plants can do.
Definitely. And, you know, without healthy soils, you'll struggle to have healthy plants as well. So an area where I guess the carbon sequestration sort of potential of like coastal ecosystems has been definitely something that's been getting increasing attention globally, but also in Australia. So mapping and preserving coastal ecosystems and their soils and their vegetation. So I'm talking about salt marshes, mangroves, seagrass ecosystems, right? So all those blue carbon ecosystems, they definitely have a lot of potential to store carbon. That's, you know, sometimes even been contrasted with terrestrial carbon. They're actually more efficient. And so that's why we should invest probably more resources into studying them and conserving them, because surprisingly, we still know very little about the sort of microbial ecology of these systems as well, and how much, you know, that carbon is then being utilized by different parts or components of that ecosystem. So great, great discussion here. We are getting some questions from people as well, but we might sort of leave them towards the end and continue with the other questions we've received before. So the next question we had is, I'm interested in how ecosystem function should be measured and monitored with respect to coal mine rehabilitation and establishment of native ecosystem outcome. So coming back to that sort of restoration ecology and, you know, what functions should we be monitoring to, you know, ensure a successful rehabilitation?
Christina, you probably have a lot of input on this.
Yeah, so I've done a little bit of work on that. So I suppose, yes, with ecosystem functions, we, I guess, have to first map out what is it that we want to achieve with the vegetation that we are replanting. You know, whether it's just presence absence, having some vegetation better than no vegetation, or are we actually looking for having vegetation that will provide ecosystem services? So for example, carbon sequestration, or, you know, remediation from pollution, for example, or, you know, even pollinators above ground. So ideally, of course, once, you know, the mine is closed or there's a need to rehabilitate, we want to ensure we get as many functions as possible, but that's not always very easy to achieve, of course, and it's a very long-term process, especially when the below ground has been, severely disturbed. All those fungal networks and mats are disturbed. We see a lot of sort of bacteria-dominated communities that are not really possibly, in very much of those symbiotic associations with plants. So I think an important factor there would be to promote plant communities that would facilitate re-establishment of those plant fungal communities. So arbuscular mycorrhizal fungi, ectomycorrhizal fungi. And in terms of the monitoring, it's probably, again, depending, we have to be informed by the ecosystem processes. So it needs to be like a mind map. What are we wanting to achieve? And then we can have targeted solutions. So for example, if we're interested in carbon sequestration, we could be monitoring carbon content over time and seeing whether that actually is matching what we're predicting, or there is some loss of carbon that we haven't accounted for in the soil. Of course, the typical ones are the insect and plant surveys, but also a habitat for, small mammals and digging mammals who are, turning around all that soil and spreading the fungi and contributing to that, healthier ecosystems. So that's my take on this, probably, in terms of this question.
Anna,
do you have thoughts based on your work?
Yeah, so I think that it's also important to monitor what the changes. So Christina, you've talked very beautifully about, monitoring the vegetation and the insects and stuff. I think monitoring what's happening in the soil is also important. And
yeah, absolutely.
My background would tend to be in monitoring the fungi particularly, but also bacteria. One of the challenges that we have in that kind of space is that we can so the main way that we'd be monitoring those fungi and bacteria is through molecular tests generally. And that usually gives us some approximation of the taxonomic groups that are in the soil, and then we link those taxonomic groups to function. I mean, that's the kind of standard way people would do things. But in a lot of cases, I feel like those links to function are a little bit, not tenuous, but they're not as certain as they could be. And so I think that there are more molecular tools that are coming out, like shotgun sequencing and things that might give us better understanding of what the functions are in those soil. But that link with function, I think, is really key. And I think that's something we need to keep working on, because once we can understand that function better, then we can understand how that soil is contributing to the ecosystem health. So I think that's still a space where we're working on really hard, and I think everyone who works in soil biology just wishes there was a magic easy answer because that's what we really want to know. I mean, the taxonomy is important, but the function is, to me, the key bit there. I don't know, Jeff, if you have any more thoughts about that.
I'm reflecting on some work that we did. So we did some work with the restoration science team in New South Wales government. Where they were looking at how restoration had progressed on sites in the Hunter. So they had a whole bunch of former mine sites of different ages, and then they had reference sites that they were comparing them to. And so it was going back to the idea of interdisciplinarity. It was a really interdisciplinary study, and so they were doing all of the above ground measurements, looking at structure, function, and composition above ground. And then we also had a bunch of measurements where we were looking at those same classes of variables below ground. The functional measurements were largely associated with enzyme activities. I believe that was the main thing, looking at carbon, nitrogen, and phosphorus cycling. And they were valuable for giving us an idea about what was happening nutrient-wise at each site, but it wasn't really all that valuable from a monitoring perspective. The most informative measures for getting an understanding of how those reference sites were, those sites undergoing restoration compared to the reference sites related to soil carbon. Not the total amount of soil carbon, but the fractions of soil carbon. So the resistant fraction and the particulate fraction. And then microbial biomass. that was the other like key measure below ground. And so in that study, we did that using phospholipid fatty acids to give us an idea of that. And then fungal diversity as well. Let's champion fungi. Fungal diversity as well was important at separating those two, looking at the richness of OTUs. But as part of that study, it was quite cool. They also wanted us to account for the costs of doing these analyses. And so in terms of the most cost-effective indicators, you had the fractions of carbon, and microbial biomass was still quite valuable, but the cost of generating the fungal diversity data reduced its value as a way to kind of predict recovery. But none of the functional variables really came out in those analyses. And so maybe maybe that means that we're not measuring the right functional variables there.
Yes, the sort of prohibitive cost of these analyses is something we've identified as well last year in a paper we published in Restoration Ecology. That was more about the restoration, exactly increasing that interdisciplinarity between plant soil ecologists, microbiologists, managers on the ground who are trying to restore, any ecosystem. So these principles apply to any ecosystem, but that particular paper was focusing on the coastal systems. Yes, so the cost of these analyses, and also analysis of the data is not, it's not something you can sort of whip up in one evening. It takes a bit of a time and a know-how and expertise. But again, I think this is just another sort of call for working together and establishing those links with on-ground people so we can translate some of that research we're doing on a daily basis as well. Another thing I just wanted to mention as well, some of the work we did in Western Australia, when we looked at some of the soil chronosequence comparing to the reference site post-mining, that was a sand mine operation. But that was interesting outcomes we found is that, often in restoration, in mining restoration, we use, those pioneer species like legumes, so acacia species, because they're fast growing, they're doing all the right things to, save cost. But we found, interestingly, that some of those stockpiled soils that they've sort of, dug out the sort of pit and stored the soil next to the mine, some of these older soils that were left there that, were kept for the purpose of restoring the operation once it's finished, we actually found that some of the bioassay species, legumes, didn't grow at all well in those soils, and then subsequently using this next generation, like high throughput sequencing methods like Anna was mentioning as well, we were actually able to identify that those soils lacked some of those key nitrogen fixing bacteria that are very important for legumes to sort of establish and grow and succeed. So that was really interesting that we were sort of able to, this is it doesn't infer causation, it's a correlation, but it was definitely interesting to see, because we looked at the fungal communities in the roots as well, and we found that they were there, but what was missing were the nitrogen fixing bacterial communities in those very old stockpiled soils. And then the acacia biomass was also the lowest in those older soils. So there was this it seemed to make sense that could have been one of the issues why acacia didn't grow so well. So if we went to then and restore these systems, we might not, have that success with acacia regenerating. So something definitely, that could be used to inform more, inoculating the soil potentially with, rhizobial communities that, your plant species will require potentially.
Christina,
yes.
Question for you now. So do you think would be then better to still use native species of the area like acacias, which are not going very well, or maybe choose plant species which are not native or maybe not endemic of the area that we know grow very well in very soil with high concentration of heavy metals maybe? Because this is the approach that we use in Europe.
Yes. Yes, it's a good question. My background is in invasion ecology, invasive plant ecology. So my immediate answer is probably no, but it depends what plants you're talking about, because the good thing about legumes in Australia or acacias, there's over 1,000 species in that family. So there are and with acacias, this could be a separate webinar, but with acacias, there are, specialist hosts and generalist hosts. So I think it's just a matter of fine tuning and testing, a species that possibly is native to the area, because we definitely don't want to introduce a species that later on becomes problematic and escapes those, constricted areas, becomes a problem in the state. But I think it's just, the whole question of plant host specificity towards particular microbial groups is another thing that we need to consider, I think, in these sorts of scenarios of restoration.
Yeah, can I just add to that as well? I think that I don't work much in restoration now, but I know that previously, like the work that, say, Alcoa did, for example, in their restoration, when originally in their restoration in the kind of 60s and 70s, they were using Eastern States eucalypts and stuff, and they found, because they grew quickly and they seemed easy to access and things, and then they found that, by the 80s, they were all starting to die off. So they'd grown very well initially, but they weren't coping very well in the long term with a lot of those different climatic conditions, different soils, and probably different microbes as well. And so there's been a real push then to start using those native, native to the area. And so they've had to actually, Alcoa themselves have had to replant a lot of that early restoration because it just wasn't very successful ultimately, even though it looked very good to start with. But I think that there's also a question then now about how we cope with potential climate change too. And, so the mantra for a long time seems to me have been, you use the species that are native to the area and you try and keep the provenances the same and all those kind of things. But now with climate change, I wonder about whether we need to rethink those things a little bit too and think about what the future climate's going to be like. But the soil will still then be the same. So there's a bit of a tussle there, I guess.
Yes, the whole, climate adaptability of plants and their microbial partners is definitely an intriguing area of research that we're still getting results in, I suppose. And, there's definitely some work published on that. But we're seeing, I think some early studies are already suggesting those functional shifts in fungal communities. I think, Anna, some of your work and our work as well, showing in those sort of experimental settings where you have those rainout shelters and temperature chambers. We are actually, one of our study looked at that in Western Australia, which is a biodiversity hotspot that has unique endemic species. And so we looked at some of the fungal communities in those treatment plots, and we found that we were losing some of fungal groups, functional groups that are, typically associated with Epacridaceae species in this family, and also sort of a shift to more pathogenic or saprotrophic fungal communities under those, increased temperature conditions. And I think this has been shown across a few other places in the world as well, not just here. So it's really, it's a worry, but it is a sort of, it's also intriguing to see that consistent response that we're seeing across those climate change treatments.
Yeah, I mean, that's really similar to what we found in other studies. So that study that we did in Western Australia was one where we had that manipulated experiment. Then we've also done those other studies where you've had, wildfires and drought and things like that. And the fungal responses seem to be moving in similar directions, like a decrease in those symbiotic mycorrhizal species and increases in some of the other functional groups like the saprotrophs and the pathogens. So yeah, I guess, the patterns seem to be consistent, but it's quite disturbing in terms of what the impact might be on those ecosystems.
Yes, definitely. And you mentioned the keyword there that just reminded me, an area that I feel we have understudied is the response of microbial communities to fire in Australia and other fire-prone ecosystems. At least, I mean, correct me if I'm wrong, panelists, but I feel this is something that there are some research coming out from temperate systems, I think from New South Wales, but we need more of that research because, again, that would inform our, ability to adapt and sort of prepare plant microbial communities to those, altered fire regimes. If we can, I don't know if we can do that, but definitely we lack research there, I think. What do you guys think, Jeff and Gian?
I just want to highlight a paper that I believe was published last week, I think,
Oh,
in
okay.
molecular ecology. So Eleonora Agidi, who's
Yep.
in the audience, with Ellie Bowd.
Yes.
Yeah.
Mm-hmm.
So that's so Eleonora's done has published a few papers looking
Yep.
at fungal responses to fire.
That's the ones I was actually thinking.
Yeah.
Thinking
Yeah.
in my head. Yeah.
Yeah.
I know there's more research coming. I think there are, I know of PhD projects from like Tasmania and other parts of Australia that are coming up. But yeah, we're staying tuned for those results and seeing, you know, if there's a consistency across Australia or not, or it's very region specific, which you could expect due to sort of variable environmental conditions as well in these different areas of Australia.
Yeah.
Yeah, one of the interesting things that we've noticed, and I don't want to preempt this, but we've been doing work looking at, you know, disturbance, fire and drought and other responses in ectomycorrhizal dominated forest areas, but we've also been doing in Banksia woodlands, which are not ectomycorrhizal dominated. You know, Banksias are not mycorrhizal, and there are some eucalypts and other, you know, myrtaceae in there that are mycorrhizal, but the dominant tree species are not, and we're seeing quite different responses in those non-mycorrhizal woodlands to fire and to you know, herbicide use and other different types of disturbances. So I think that could be really interesting too. And I think I've seen some other work that's come out of some of the research group at Kings Park in UWA that have done some really interesting stuff looking at communities in the Pilbara and other areas that have shown that they're not responding in the way we expect to. And I think that's really interesting because a lot of that fire response work and stuff has happened in forest areas that are dominated by ectomycorrhizal species. Yeah. I think that's really going to be quite fascinating to see how that plays out too, though we may not expect everything to respond in the same way.
Even in those non-mycorrhizal woodlands that you're working in, those are quite open. I would imagine that after fire, there's quite a grassy, the grasses recover quite quickly.
Not so many grasses in the Banksia woodlands. Yeah, it's quite a different kind of ecosystem to the other woodland types that we get. Yeah. So it's, you know, something a bit different again and maybe quite specific. It's a bit more like a low heathland or something in the understory, if you like. Okay. Yeah.
Okay.
So yeah.
So maybe some ericoids.
Yeah.
Yeah. We've actually had a comment from one of the members, attendees, Mercedes Ondik, who said that they have a paper coming out next week that looked into bacterial and fungal responses to a high severity fire in Kangaroo Island. So there you go. We'll keep our eyes peeled for that paper. Thanks for that comment as well. Excellent. So, okay, so we've sort of, it seems that just organically, the first part of our webinar was sort of focused on interdisciplinarity, applied sides of plant soil ecology. We also got quite a few questions that came through about slightly sort of different side of plant soil ecology. So we might address them as well before we're running out of time. And we've got a few questions in the chat that we hopefully can get to as well. So we've got some questions from someone who's studying blue carbon storage in estuarine mangroves and the role of invertebrates in decomposing specific types of above ground biomass. So I can just disclaim immediately that I don't know anything about invertebrates and their role in that whole process. I acknowledge they're important. So I don't know, you know, how well I can definitely address these questions, but we can definitely cover this topic and some of the questions that came through with that. So the one we can start off is, how can senescent above ground biomass containing tannins assist in the degradation and the nutrient cycling process? As most of the literature provides evidence on the contrary, on how the tannins discourage herbivory, is there a phenomenon where the opposite occurs? So I hope that made sense how I read out this question. But if anyone maybe has experience with invertebrates and that this side of things with the in those systems, or any systems really, it doesn't matter.
We've done a little bit of litter decomposition work here in eucalypt woodlands, and those chemically, those leaves are quite complex and tough for herbivores to eat, and they do decompose relatively slowly.
Mm-hmm.
Yeah, so I would imagine that in these in the systems that the person asking the question are like, I could imagine that it would be similar there, that you'd end up with this relatively slow release of nutrients and slow breaking down of the carbon in that material.
Mm-hmm. Yes, I agree. So I think, yes, as you say, Jeff, it doesn't really matter which ecosystem it is. I mean, mangrove and blue carbon ecosystem, of course, is a bit different to the rest of terrestrial ecosystems, just because of the osmotic stress, the salinity, there's all these inundations. So they're definitely affecting the processes and possibly slowing them down, or maybe even speeding up. And in some areas, it again depends. But yeah, as the biomass decomposes, of course, it always releases all those chemicals, but also the carbon that then can be used, for other plants. But I think I just would like to preface, overall, having done a bit of research in blue carbon ecosystems, we know a lot about the biogeochemical side of things in those systems, and the cycling of nutrients, but the microbial, and I think in invertebrates, I'm not even going to try and answer that question, but the microbial side of things and the decomposition and these processes, we still don't know enough. And I think this is definitely an area that I feel it's getting more traction and more is coming out. But it's something that we're still not too sure of. The same with, say, fungal communities. There was another question about, the fungal communities on mangroves and how in the warming climate will that affect the litter fall and just, I guess, the plant responses. So again, from what I know, we don't know too much, because now in blue carbon system, the fungal communities are sort of a mix of marine fungi and terrestrial fungi. We recently did a review where we looked at, how much do we know about the fungal communities just generally in those systems, in blue carbon ecosystems. And we don't know much, like the endophytes. What are the endophytes doing, either inside the above ground parts or the below ground parts? It's a bit of a mystery. So I think this is an area where we could expand and work more towards as well. There was one question, though, that I think the panel probably would maybe have more comment on, is how reliable are the soil organic matter measurements from past studies using robust methods like weight loss on ignition? So I think some of the panel members have experience doing these sorts of analysis. Did you have a comment on this and how reliable it is compared to current methods, possibly?
I can start this one if you like. So we use loss on ignition as a simple and quick method just to measure carbon often. And I don't feel like it's giving the complete answer. I think that often it doesn't account for the different carbon fractions and how they might be contributing to different parts of the soil process. And often with often with the loss on ignition work that we've been doing, we get answers that we're not really expecting, that don't seem to reflect what we would be seeing in those ecosystems. So for example, we've been doing some work in regenerative agriculture, and we've got one pilot study that we've been doing. We've got a really nice setup where we've got some regenerative farms right next to some conventionally managed farms. And so, they're adjacent paddocks. And so we've been taking samples over a number of years now, and using a simple loss on ignition measure to look at carbon seems to indicate, at least from these pilot farms, that we've got more organic matter in the conventional farms, which is not what we would have necessarily expected, given that some of those other farms have been organic for a long time. And so by then digging into the different fractions of carbon that you've got in those soils and things, and looking a little bit more closely, you get a much more complex and a much more interesting answer, I think. So at a superficial level, I think it's a good start, but I think that it's not giving us the whole story about what's happening in terms of that carbon cycling in the soil. Gian, did you have something else you wanted to say about that too?
Oh, has the loss of ignition, it's considered like it's easy to do. I found that it's also so easy to mess up or to not keep standardized. Two furnaces, different brand, can give different results. The time that you use to burn the material changes the results. The temperature changes the results. And sometimes people just put, let's just put 800 degrees, and the results that you get is everything that is just burning is not just organic matter. There is a lot of stuff in there. You use a temperature that is too low, you didn't burn enough organic carbon. So, and like you can repeat, and if you just 1 year forget, did I put 350 degrees for 1 or 2 hours? Let's just put 1, it's going to be the same. No. Spoiler alert, is not going to be the same. And yeah, that's each lab does a different way, and I don't think it's easily comparable. So yeah, I am a fan of loss of ignition, don't get me wrong, but we should like find a way to just use the same way everywhere, because I think like that's going to be an issue.
Gian, that makes me think that we need a methods paper, if there isn't one already, that sort of outlines, okay, how maybe we should be doing this to kind of ensure standardization. This is something I haven't personally done, so I'm not an expert on this at all, but just kind of listening to the discussion, maybe there is a scope to think of some sort of contribution to call for that, or summarize those, known methods and the results, and see, even though they are variable results, are they still within the standard errors, or, the error, or are they giving us totally different and erroneous results, which is a problem, of course.
And I guess it depends on what your objective is too.
Yeah.
If your objective is just to compare the results within a particular study that you're doing, and you're consistent in how you approach that, then it's probably fine in terms of standardized approach. But if you're wanting to compare between different studies or across years when you might forget your methodology, then that becomes really problematic.
Yes.
There is like a new method where they use a machine, it's called LECO, L-E-C-O, and they check like the carbon dioxide that is produced. So they do like gas chromatography. Downside is bloody expensive. You need to buy like a multi-million-dollar machine to make that. So there's always like loss of ignition, it's so easy and it's relatively cheap. Other method, you need to buy like a super expensive machine to get more direct and robust results. But like, is this amount of money worth? That's a good question, which I have no idea. I have no money, so we'll keep doing loss of ignition.
Yes, it's a challenge, isn't it, always? And it's also just what's available at your institution or your collaborators or your supervisors. So there's only that much you can do. But I think Anna's point is excellent, is that whatever you do, be consistent across at least your studies or your experiments. And another thing is, of course, the reproducibility in papers. Just if you have enough detail that we can at least see how it's been done, then, that gives us a good baseline to compare different methods and the results as well. Sorry, Jeff, I cut you in there. Did you have a comment?
You made the point that I was you document what you do.
Yeah.
So that it's reproducible.
Yes, very important. Excellent. Well, we have about 10 minutes left in this webinar, but we can go a little bit over if the panelists are okay with that, and the attendees, because we do have quite a few questions and comments in our Q&A and chat. So I might just start with addressing those. We've got a comment from someone saying, so glad to see my question on weight loss on ignition is being answered. Currently testing out different temperatures on my samples in order to standardize my samples. There you go. So good to hear that. The other comments, I'm just going to maybe go from the beginning and see. Okay, we've got a question from Adriana asking, in the consultancy area, invertebrate and any plant-soil interaction is considered in standard impact assessment for development or revegetation. Does anybody have experience or knowledge of that sort of approach? So I'm assuming the approach for these assessments.
So in New South Wales, there are standardized assessment approaches. So it's called the biodiversity assessment method. And it is, there's a manual associated with it so that consultants can follow that while they're doing their assessments. But to my knowledge, invertebrates and plant soil interactions aren't included in that, although I could be wrong.
Yeah, my experience is in Queensland. I think it's the same. We do have a sort of general framework, biodiversity assessment. I'm yet to look into it actually for my teaching purposes as well, but I don't remember from the top of my head if the sort of soil side is taken into account at all. I think it's all sort of plants, maybe, you know, mammals or, but this is definitely probably something coming back to that discussion on, you know, biodiversity of credits and things, including, you know, the below ground, some sort of metrics to account for those as well.
I wonder if the Global Soil Biodiversity Initiative, is what they're called, might include some of those things now. I haven't looked at them recently, but that's an international body who are looking at soil biodiversity. And I imagine that's the kind of thing that they would be developing at some level, I would hope. About the invertebrate monitoring though, like just putting on a different one of my hats for a second, there's been some work in iDNA, so invertebrate environmental DNA, that's come out of Curtin. So Christina Fernandez did her PhD looking at invertebrate DNA for monitoring restoration success. So that wasn't specifically soil invertebrates, that was general invertebrates, but she did do quite a lot of interesting work there, looking at how you might use molecular tools to monitor invertebrate diversity in restoration space. So not necessarily a great option for consultants because again, it's quite expensive, but another tool that we might be able to use in the future. Thanks,
Anna. Yeah, so Adriana further commented that the BAM or the Biodiversity Assessment Manual does not have anything on including invertebrates or plant-soil interaction. So it focuses on plant cover and threatened species, mainly vertebrates and few invertebrates like land snails. Yeah, so this is what we were expecting. So maybe again, something we need to be thinking collectively as plant soil researchers in Australia as well to include that. But I think Anna's comment on the GBIF, the Global Biodiversity Institution, they probably definitely have manuals for assessing, at least, you know, generically maybe the soil biodiversity that could be applied or maybe adopted to Australia. Excellent. Let's see. We've got a few. We've got a next question from Carl regarding biodiversity credits. Is there a precedent in rewarding soil biodiversity? I'd imagine it's hard to quantify both microbiota and macrobiota diversity on the ground compared to above ground. Which kind of relates to our previous question again. So having a method would be, first of all, great. And I think as all these things, when they start off, it's more, you know, superficial, generic initially, but then again, depending on the purpose or the question, what are we trying to achieve, you know, they might need to be fine-tuned. But so far, I don't know that there is any reward for biodiversity, soil biodiversity in Australia.
Yeah, not to my knowledge.
Yeah. Next question is from Liz. Regarding interdisciplinarity and collaborations, are there guidelines for non-soil people on how to collect and store soil samples for later collaboration? And as a specific example, can soil samples that were collected months ago and dried still be analyzed for soil fungi or microbes? I'll let someone. Gian, have you collected samples recently? Or
so I use one book that is like phenomenal. It's called like the Canadian Soil Sample Methods, which is like my Bible that I follow like this. Funny thing, each soil scientist will use a different method in the end. So certain people would prefer using like just grab some soil, put in a paper bag, and then air dry for 2 days. Certain others would say, no, don't air dry because you might contaminate your soil, just oven dry. Certain others would say, no, that's not good anyway. You need to get them frozen. So I think it depends if you want to collaborate to just before that decide on a unanimous way to store the samples, and that's it. I know that for microbes, I think you have to freeze your samples. For chemical and physical analysis, it's not necessary because I never done it. So let's hope that it's still okay.
Yes.
That would certainly be my recommended approach, would be to freeze your samples as quickly as possible, because there can be some changes in the microbial biomass and things in response to the changing conditions once you've collected them. So that's what we tend to do, is to always put them in the fridge. So if they've been sitting around for months, even if they're dried, I would still be hesitant to use them for next generation sequencing or, any kind of molecular analysis. But that's not to say that they wouldn't be completely, they could be used for something else. I know, Jeff, you've done quite a lot of biomass work, haven't you? So would that be appropriate?
So biomass is, you do need to freeze it because a lot of that biomass is in bacterial cells or it's in fungal hyphae, and those, once they're disturbed, the signatures associated with that biomass that we use to measure it start to break down. If you're looking at aspects where the structures are a bit more resistant, so like fungal spores, those can persist in air-dried soil samples. There's the Australian Microbiome Initiative it's a pretty amazing data set that's available online through Bioplatforms Australia. It was funding from Bioplatforms Australia. A lot of it came from CSIRO. They have included hundreds of samples, air-dried soil samples that they had received from Geoscience Australia. I haven't looked at them in too much depth as a comparison between the samples that were frozen. They will be different, but they were still able to get quite a bit of good data from those samples. So just storage method is important. It's important in terms of how you interpret the data, but the data can still be valuable.
Yeah, I'm glad you mentioned that Bioplatforms Australia, because I think that relates back to some of the things we were talking about before. I mean, they've sampled quite extensively across Australia and in lots of different ecosystem types, and that data is publicly available. Mm-hmm. So some of that's data that we have. The reason I just remembered was that when we were doing biomass, Jeff, we were doing ergosterol measures, and you can do that on air-dried soil.
Okay.
Memory, because that's mostly related. That's fungal biomass rather than bacterial, and so it's a little bit more resistant to degradation.
Okay.
Yes, I agree. Yeah, I agree with everything that's been said. And just coming back to the to Liz asking that question again, I think it's the answer will be very much informed by what are you trying to get out of those soil samples? What's the aim? Without going into too much detail, if you're looking at the DNA of microbial communities, or you want to have a proxy for richness or diversity in those soils, then DNA degrades with time. So as a standard, we put the soil sample we've collected, ideally not in a wet conditions, because wet conditions will promote those pathogens in the soil proliferating. So first of all, you want to sample on a sort of normal sunny day if possible. And usually we put them in sterile tubes and in -20 freezer if that's available to you, but -10 is better than fridge. But even in the fridge, they could be for a while. So again, it depends. The storage, as everyone has said, the storage and handling is important, and sampling as well, not to cross-contaminate between, say, sites. So wearing gloves, using bleach to clean your tools and so on. But again, the question will inform how exactly you would have to store. For example, I've done some RNA work, which is when you're actually trying to extract RNA to look at what we call sort of the active microbial community at the time of sampling. Because you have to remember, DNA is everything in the soil that has ever existed at that site. So with RNA, we had to put it in the liquid nitrogen in the field while being out on a boat in the marshes of Louisiana. Crazy stuff like that. Didn't work very well. Don't want to do RNA work again, but just gives you an idea that it very much, the question will inform, and yeah, the people who are collaborating with, would ideally know and suggest the best method. I think that's my answer for that question. We have another question from Sadia, who's asking, to know the functional diversity of fungi and bacteria due to climate change, what should we measure? Is Illumina MySec able to answer the functional diversity question? Anna, would you like to start off?
I'm just smiling. This comes back to what we were talking about earlier, I think, about so Illumina, so MySec really is looking at the taxonomic diversity within your samples, depending on how you're doing it, but generally the high throughput sequencing tools that we use would be looking at taxonomic diversity. And then where we know enough about the taxa that we're working with, we can link that to function. And there are a number of different databases that allow you to do that. So that does give you a bit of a proxy for function, I suppose. Like it's, there are those functional databases are, say, a lot more robust for areas where the fungal or bacterial diversity have been a lot better studied. So Northern Hemisphere particularly, I think that the functional database is a lot more robust. But in Australia, I think it's getting better, but it's still, I know when we first started using Funguild and some of those functional databases about, I don't know, maybe 10 years ago or however long ago it was, that they had almost no Australian species in them or no Australian genera even. And so it was really difficult to infer anything about function using those databases, but it's gotten a lot better in the last 10 years or so. Yeah, so I mean, that's my experience. We kind of use that to infer function. It's not perfect, but it's one of the better proxies that we have now. There are other sequencing tools like shotgun sequencing where you're looking at functional genes. Yeah, I've done a little bit of that and it's really interesting, but I still don't think we know enough about which genes are related to which functions as well. And particularly for like the mycorrhizal genes, there's lots and lots of different genes that, there's not just, one or two mycorrhizal genes that we can target. There's lots and lots of different ones. And so that makes it quite challenging to answer those questions about function too. Jeff, do you have some thoughts about this as well?
No, I guess I'd just add that some of those other sequencing methods, so using shotgun metagenomics, there's a lot of value in them. It gives you a ton of data. As you said, Anna, you're kind of taking it can be a bit challenging to interpret what those results mean and how the quality of those data. But it does allow you to do some pretty fantastic things. Like if you have enough of it, you can reconstruct entire genomes of mostly bacteria that are available in the soil samples. It's tricky from soil samples, but yeah, when you're working in soil, it's extremely challenging because the diversity is so high. There's so much environmental heterogeneity. Eventually we might be able to get there, but really we're largely limited to fairly simple soil systems for being able to do stuff like that.
And I think from my end, just to clarify for Sadia, just in case, so the MySec results or output will not give you functional data as an output. Obviously, it will give you the sequences that then you can go, check for the taxonomy file, and then you'll get the names, and then those output files you can use in things like Fungal that's been mentioned to try, that basically just matches from other studies, whether that fungi has been assigned a function, it's doing something. It could have been a field study, it could have been a pot experiment. So I think that's a really important understanding that needs to happen. And same for bacteria. So for bacteria, something like pie crust could be used to get data on the functional diversity of your bacterial community. So there might be some methanogenesis involved bacteria or other. So MySec, I suppose that's what I've done at least, and feel free to correct me, anyone. But yeah, you get the data that you can then feed into other platforms to get that functional data in return. And in terms of the climate change, I think, again, if you're interested in microbial responses to climate change, then so for example, something that I know Anna and myself have done is we have those experimental field plots where we have simulated the climate change conditions for that particular area, and then we have also our reference or baseline site where we sampled as well, and from those experimental plots, and then we can compare the sort of those functional changes. So we can possibly, I don't know, send you papers as idea if you're interested in to sort of get more information on these sorts of experiments where testing on climate change effects. The next question, so we just got a few more questions and then we'll wrap up. Another question from Alison. Has any of the panel considered moss as the plant side of the soil-plant interactions?
I haven't. I don't know if anyone else has.
I haven't either, but I think it's a great suggestion. I love
really,
all those small plantings.
It's really cool. Like there's there are groups elsewhere looking at moss mycorrhizal interactions kind of as a way to understand how that symbiosis might have worked early on in plant evolution.
Mm-hmm.
But yeah, I haven't done that myself.
I haven't either, so can't comment much there as well, but I agree it would be really interesting, or I'd like to know more anyway. Gian, do you know of anyone who might have worked on moss? And no.
I know that David Eldridge worked with mosses. Max Mullen Cooper, they work with mosses too. They are great at ID mosses. I can't imagine doing myself and soil interactions on mosses, because I just think how particular you have to go. Mosses don't have roots, so you need to just focus on the first 2 centimeters of soil. I have a shaky hand. I don't think I can try to collect soil just at 2 centimeters. That's why I go 10, 15, and that's why there is not much done, because it takes a lot of time. But Alison, go for it. That's where you have.
There's a comment as well that there is, Katie Field's group is looking into bryophytes and AM fungi. So I believe Katie Field is based in UK, if I'm not mistaken. Yes. Okay, we've just got final question and we will have to wrap up. So last question is from Jack. On the restoration side of things, how useful do you think reference restored soil comparisons are, given both the time required for rehabilitation and the potential for restored site to reach alternative stable states? Might be a little off topic, so feel free to ignore. That's okay. We haven't. It's a good question to finish off, I think, on the sort of applied side again of plant soil ecology.
I guess its value is in that you need a target,
and
it's you need a target for that particular climate zone, you need a target for that particular soil system, you need a target for that vegetation type. And a reference site allows you to identify that target, whether that target is achievable. That's another question. But you do need that target. Maybe there are alternative ways to come up with that. Maybe some of the other panel members have thoughts on that.
Yeah. Useful, yes, it is useful, but like using that as what this ecosystem will have to be, I think it's wrong. I don't think an ecosystem like a mine site that's been so messed up will ever be able, even 100 years, to reach that point. Like, even with all the efforts in the world, it should be a guide. This is the path we should aim to, not this is what it will have to be, because I think that's unachievable. It's good to have as a reference, not as a target.
I agree with both those things. I think with a lot of the soil microbial work we need to have some kind of comparison, because we don't know enough about everything, all the different components of that soil ecosystem and what they all do. Then it's always really important to have some kind of comparison. And I think that those reference sites are a good comparison to give you some idea of what different functions might have been important in that ecosystem, even if you don't end up with exactly the same species, but you might have similar kinds of functions, or even, slightly different functions. It gives you a good way to think about what you might be aiming for, but what might have been important in that ecosystem, previously. So yeah, I think we're, as soil ecologists, pretty aware of the fact that those reference sites may not be achievable, but yeah, I do think they're really valuable in giving you some idea of what to aim for.
Yes, I think I agree. We always need a baseline and a reference point. So it might not always be ideal, but we need to compare sort of the modified conditions to the pre-existing conditions. So definitely need that as well. So baseline is always there, or control or baseline, whatever you want to call it. Well, I think we've gone a little bit over time. Apologies to the panel members and also the attendees, but we did have a very stimulating discussion that it was just shame to cut it off and not address the questions we got from the audience. So thanks everyone for staying on. Apologies for going a little bit over time. I hope everyone enjoyed it. Get in touch, please, with us individually or via socials if you want to. We are on Twitter as a research chapter. If you had some questions and you wanted to maybe get some of those references and resources we have mentioned, we would be happy to share. I know I would be. And otherwise, yeah, thanks everyone, and we'll see you somewhere else in the soil sphere. Bye.